Welcome back, guys. We have just discussed about the RNA interference, or what is the concept of RNAi. Now in this video we'll be talking about the continuation of this series of RNA biology, and we'll be talking about a type of RNA interfering system which is called as microRNA or miRNA. So let's talk about this. So what is microRNA? Like other type of RNA interfering system, it is also generated from a double-stranded RNA inside the cell. So even inside the cell, if by any means there is a double-stranded RNA synthesized, in that case that double-stranded RNA could be of three different types: siRNA, shRNA or miRNA. So there are differences in their structure and the number of nucleotides they possess. Now miRNA is generally produced inside. So let's start with the double-stranded RNA thing here. Double-stranded RNA thing. So we need to have this double-stranded RNA to have any activity of RNA interference. So let's say we have double-stranded RNA. Now the question is there, how we can get a double-stranded RNA? Now this double-stranded RNA that we have in this case of miRNA, which is also called, in miRNA, that double-stranded RNA will look something like this. Like this. So what it has actually, it have stem loop structure forming like that. So let's say this is the stem loop structure. It looks something like this. This is how miRNA looks like. This is the miRNA. It is a double-stranded RNA, of course. Now if you see this, you can have a fair idea that this is produced from a single-stranded RNA. How? Because a single-stranded RNA is pretty long and they have kind of complementarity between the bases. As you know, this will act like a Velcro. We all know Velcro is. So Velcro can actually stick to each other. If we have a long Velcro, we just put it together, it will somehow fold and attach with each other. Similar things, because the bases are always have the eagerness to bind with each other by hydrogen bonds, right? So if these are short nucleotides, it's pretty difficult for them to fold back and adhere. But if this is a long nucleotide sequence, in that case, it is a possibility to actually produce some structure like this. So it is produced from a single-stranded RNA. For example, that single-stranded RNA was like that. And that single-stranded RNA is now actually converted to this double-stranded RNA. But there is an important feature of this single-stranded RNA, and that feature is, remember, is the sequence complementarity. Sequence complementarity is there. Say inside the cell, this kind of sequence complementarity is formed. Due to the sequence complementarity, it folds back to form a hairpin-like structure, which is ultimately called as a miRNA there. And this type of RNA is generally produced, again you all know, the RNA is usually produced by RNA polymerase II. You all know that. They are produced by RNA polymerase II. So once they produce this miRNA, it is termed as miRNA, not pre. It is termed as miRNA. So this type of RNA is produced inside. And the normal production process we've discussed is completely endogenous. That means the process here to produce the miRNA is inside the cell. We are not providing this miRNA from outside, because in all this RNA interference, there is always a room for us to deliver that miRNA or RNA interfering compound from outside by injection, microinjections, right? So this is the endogenous origin of miRNA there, and it is called tri-miRNA. So once the pri-miRNA is there, they form loops like that and bond in between, then again loop bulge. The structure is like that. So double-stranded RNA is produced. Now that double-stranded RNA will obviously now do the damage, right? So how will it do the damage? The situation now, this pri-miRNA is now inside the nucleus, and everything we are talking about is inside the nucleus. I'm going to draw a barrier when we will talk about inside the cytoplasm. So it is inside the nucleus. And there is an enzyme inside the nucleus that is called as Drosha. So it is called as D-R-O-S-H-A, Drosha. So the Drosha and it's DGCR8, this is another enzyme. So Drosha and DGCR8 combine together and they act on this miRNA. And remember, these miRNAs are kind of 100 to 120 nucleotides long. And this Drosha and DGCR8 act on this miRNA. It will cut some region of this miRNA. For example, it cut from this particular location. So it makes this miRNA shorter. And the miRNA it is producing then, it's called as, it is termed as, so let me draw it for you here, like this. Like that, right? And it is termed as pre-miRNA now. It was previously called as pri-miRNA, now it is called as pre-miRNA after the cleavage. And this pre-miRNA is 70 nucleotides approximately long. It's approximately 70 nucleotides, 65 to 75 nucleotides long, pre-miRNA. So once the pre-miRNA is produced, after this pre-miRNA is ready to be sailed to the cytoplasm. And this pre-miRNA is brought into the cytoplasm by the enzyme, by a protein, transmembrane protein that is present there. It is called as, it's a protein which will facilitate and actually guide this pre-miRNA to be excluded out from the nucleus into the cytoplasm. We know in the nucleus there are nuclear pore and nuclear pore complexes are there. So from that complex there is a protein which will attach to the pre-miRNA. It will guide the pre-miRNA and finally bring it to the cytoplasm. And that protein is termed as exportin. So exportin 5 actually. Exportin 5 brings it there. Exportin 5 or 5 whatever you write. It brings this pre-miRNA to the cytoplasm. So now on, whatever we'll talk about from this barrier will be in the cytoplasm. So up till this part, until this particular barrier, everything was in nucleus. After this, we'll bring it to the cytoplasm. Everything is now cytoplasm. So this is the limit of nucleus. Now everything will be cytoplasm. So a pre-miRNA is there in cytoplasm now. So once we have the pre-miRNA there, this pre-miRNA will undergo future stages of dicing. That is the cleavage. Because if you remember my first video about the RNA interference, that is a must watch video because that is the video of the series starting video, introductory video, and you never should miss an introductory video, right? So understand that introductory video and then that this mRNA should be cleaved, it will generate something, then finally they will bring it and they will do the task of degradation of target mRNA. So pre-mRNA is produced. Now they will bring upon the enzyme called dicer. Remember? They will bring the enzyme called dicer. So dicer will act here. Dicer will act here. Now dicer, what dicer will do? Dicer actually prepares this RNA to be properly activated. Right? And dicer, I have already told you, is nothing but a RNA's 3 type of enzyme. So it will actually, it is a double-stranded RNA specific endonuclease. So what it will do, so if this is the situation, let's say this was how it looks like. Let's say this is the structure of the miRNA, the pre-miRNA. And what dicer will do, dicer will actually bind with it. Dicer will actually bind with it and dicer will cleave it from a particular location, both the ends, in the opposite direction. So as you can see here, dicer is cleaving one from this end, one from this end, and again one from this end, another from this end. So ultimately what it will do, it will break down this hairpin. and it makes it a double-stranded RNA without a hairpin, right? And after this dicer processing, what we will get? We will get a structure like this, let's say, like this. So what we'll get here? We get 3' overhangs. Using dicer activity, we'll get a 3' overhang of 2 nucleotides. So if you look at here, these are the 2 nucleotide overhang, 2 nucleotide overhang at 3'. We'll get it. Also we'll get 5' monophosphate regions. So only one phosphate will be present in the 5'. So after the processing by dicer, it will break this pre-miRNA down. So it was pre-miRNA, break it down into what is called as miRNA star duplex. This is the name of this complex. miRNA star duplex. And that is what we are looking at right now. So this is the activity of dicer. And also the activity of another enzyme inside the eukaryotes. There are many other enzymes are acting all the time. But majorly, the activity is by the dicer to produce this type of structure. So once we produce this miRNA star duplex, now this is ready to be attached to the actual RNA interference complex. And remember the name of that complex? We have already studied it. The name of that complex is RISC. Loaded in RISC. And you know RISC, the full form, RNA interference complex. I mean RNA interference silencing complex, right? So RNA interference mediated silencing complex, that is the RISC. And they will load it into the RISC. And what the RISC is consisting of? The RISC is consisting of, we all know, the protein called slicer, right? And it is also consisting of argonaute. Now the name of slicer is only found in in certain examples of organism, for example C. elegans only. So in case of eukaryotes, there are other enzymes instead of this slicer. Argonaute is already present, and there is a protein called TRN6. So this is the enzyme instead of slicer with argonaute that is present there. But argonaute found to be very important and a common type of protein. It should be present irrespective of plant, animals, fungi, whatever we're looking at. This argonaute is already present there. So that makes the RISC complex complete. So now this miRNA star duplex will bind with the RISC complex. So once it is bound with the RISC complex, what the RISC complex will do? So let's say here comes the RISC complex. Let's say here is the argonaute in place, slicer or any other enzymes are in place, let's say like that, and this structure is now attached, right? So after that, what it will do? Two overhangs are there. This is the actual the miRNA, processed miRNA, and this is the slicer. This is the RISC complex is present. Now this RISC complex, major part is argonaute. This is the argonaute protein in blue color. This is the argonaute protein. Now this argonaute protein has two domains. One is called as the PV domain, which consists the activity of RNase H, which will break down the RNA, single-stranded RNA, RNase H activity. And it also have another region, which is called another domain, which is called as a PAS domain. That PAS domain is very necessary to attach and bind the backbone of a single strand of RNA. Because remember, this will no longer be active if one strand is not released. And that is the important task of RISC, to attach with one strand and release other strand. And for attachment with one particular RNA strand, they require the presence of the PAS domain. And then for the cleavage of the target mRNA, they require the RNase H activity or PV domain. Okay, it's very clear. So it consists of two domains. PAS and PV, I've told the function of them. So now this is it. Now after that, what it will do, among those two strands, it will firmly bind with one strand with the PAS domain, and that strand will be termed as, let's say this is a strand, and that strand will be termed as the guide strand, and the strand which will leave after this process will be termed as the passenger strand, because passenger always leave the place. So that is the passenger strand. So once it is denoted which strand goes where, then that RISC complex along with, let me use some other color, that RISC complex along with that guide strand will migrate to the actual target sequence of single-stranded mRNA. So let's say here we go, the single-stranded mRNA is this one, and they have a target sequence, let's say from this to this is the sequence of their target. So what it will do now, this is the whole structure of argonaute and slicer and whatever other proteins are, and it is also associated with the guide strand along with this. Actually I drawn here wrong, actually it is attached the inner side, the overhang actually present in inner side of the place. So this is the scenario. Let's say this is the scenario that we have. Now in this case, this argonaute will bring this miRNA complex. Now it becomes miRNA, actually active miRNA complex. Now that miRNA will bind with the target sequence here. It will bind with this target sequence here. This is the target mRNA. Remember, it will bind with this target mRNA here. So once it binds with this target mRNA here, it will form nucleation. Now that is called the nuclear hybrid structure. Now why? Because normally this is a pretty long nucleotide. Normally this, after this dicing and slicing, ultimately the nucleotide is kind of, let's say 15 to 20 nucleotide long. So 15 to 20 nucleotide is not very short. So they need to find the complementarity of 15 to 20 nucleotides in the target mRNA. So once they find that, it's kind of difficult to exactly find it and attached, right? So for that reason, they will actually find nucleotide complementarity, say 2 to 3 nucleotides. Let's say this 3 nucleotide here, let's say this 3 nucleotide here have a complementarity with these 3 nucleotides. So they first find a complementarity there with 3 or 4 nucleotides, and that miRNA will bind with the target with only 3 to 4 nucleotide complementarity at first. That is called the nucleation. That is the nucleation. So once the nucleation is done, then rest of the miRNA will bind or attach with the target mRNA. So once they are attached there, what they will form? They are again forming double-stranded mRNA. So they are forming the double-stranded RNA, and we know that argonaute is RNase H type of endonuclease. It will no longer tolerate the binding of double-stranded RNA. So what it will do, you know the functionality of RNase H and RNase 3. RNase 3 will bind with double-stranded RNA, cleave both the strands. But RNase H will recognize double-stranded DNA, but cleaves one RNA only, one strand only. And that's the difference. Here, it will bind with the target mRNA, and as argonaute is RNase H, I remind you, this is an RNase H type, RNase H type of enzyme, it will recognize it. So now, they will bind, so let's say this is the miRNA which is now bind. So they will recognize this double-stranded RNA, but will break down the target mRNA only. So it will cleave the target mRNA into 2 parts, right? For your understanding, I put these red dots because that is the target point and it's now broken. After the breakdown, we also have our miRNA there. This was the miRNA, right? So that is the overall process of miRNA activity. So the actual target RNA is now cleaved. So the protein that should be produced from translating that mRNA will no longer be produced inside the cell. So the cell is now free from the disease if we treat it, or cell can cause certain disease if it is malfunctioning, if it's a good protein inside the cell. So this is a very important process, guys. Remember, very small and many stages are there. They are arranged one after another, working one after another. But ultimately, the fundamental process remains the same. Double-stranded RNA produced via endogenous pathway, or we inject it from outside, exogenous pathway. Then it will be processed, it will be exported to the cytosol from nucleus by exportin 5. Then it produces the pre-miRNA, and that pre-miRNA will be cleaved and processed to produce miRNA star duplex. That is due to the activity of the enzyme dicer, which is a very important RNase 3 type of enzyme. Once that is produced with overhang structure, it will bind to the RISC complex, which is called as RNA interference silencing complex, with Argonaut and slicer or TRNC6 or TRNC6. So whatever it is, then that slicer or Argonaut, that is the whole RISC complex, will go, and that RISC complex will unwind the double-stranded RNA, release one strand which is called as passenger strand, hold on to one strand which is called as guide strand, and that this RISC complex with the guide strand will go attached to the target mRNA. The attachment of RISC strand, the guide strand with RISC to the target mRNA is based on the nucleation model. They attach with first 3 to 4 nucleotide, then they elongate the attachment, and then they cleave it. Because Argonaut is RNase H type, they recognize double-stranded RNA but breaks down only the target mRNA, not the guide mRNA there. So once this process is done, now what they can do in future, this breakdown component of mRNA can serve 3 important features now. Now that breakdown mRNA, can imagine they are having some complementary region with the guide RNA. So they can bind with the guide RNA to produce another double-stranded RNA inside the cell, which will further amplify the process of RNA interference inside the cell. Or sometimes these mRNAs are actually degraded by the cellular enzymes that is present inside. That is the most common, it is degraded. But in some other cases that can also pair with some other RNA to produce double-stranded RNA to keep doing that devastating activity that is going on by RNA interference inside the cell. It's an amplification, and positive feedback loop can be possible. On the other hand, they can also produce, again, so these are the major 2 types of situation that may arise, right? So that is the overview of miRNA. Now if you understand the overview of miRNA, it's done. You don't need to understand anymore, because the later thing we'll be talking about the siRNA or shRNA. The process will be very, very similar, because miRNA is the most complicated process. Rest of them are pretty much easier. siRNA will start from particularly this particular phase. So this all phase will not be there in siRNA or in shRNA. So we'll start from that, and rest of the process is kind of very, very similar to each other. So they are having different names, but actually they are double-stranded RNAs, and the machinery to process the double-stranded RNA to cleave a targeted mRNA is kind of similar. So that's it, guys. Keep tuning, keep watching, keep subscribing, and I hope that's helpful. Thank you.